Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm very excited to be here with Dr. Giles Yeo, who is a geneticist and author, currently professor of molecular neuroendocrinology and program leader at the MRC Metabolic Diseases Unit in Cambridge. If you're a longtime listener to this podcast, you will have heard me and Giles talk on episode 3. And I have to say public thank you to Giles for that, because there's no selfless act like going on a podcast in its first 5 episodes when you're almost guaranteed that the only listeners will be my mother and a few close friends. But my mother loved the episode. My wife really, she follows all your stuff. She's really interested in nutrition and diet. And so you got our whole family interested with your first book, Gene Eating. You've had a second book come out since then. We've had a pandemic. We've had GLP-1s come out. So we have a lot to talk about. But first of all, thank you for joining me for
That's all right. It's a different world. Oh my God, ancient history. But
yes.
No, thank you for having me back, because I clearly didn't scare you away. So that's fine.
No.
Good.
No, not at all. And we did record in person the first time, actually, which was really nice. The world has changed since then. We're on Zoom, but it's nonetheless great to have you here. Let's start with, I'd love to just get your perspective on, over the last 5 years or so, our understanding of obesity has changed. The tools that we have to treat it has changed. Genetics has continued to march on, therapeutics. I want to just get your perspective on, if you think back over the 5 years, what are the big things that have shifted in the field to set the scene a little bit for people, and how you're thinking about it now compared to a few years ago?
I mean, I think the biggest thing that we have to tackle, the 64-ton gorilla, or whatever it's called, are clearly these new GLP-1 or incretin-based therapies, right? I mean, that undoubtedly is the big change within the field and has driven other technologies and a lot of the research. And the thing about these incretin-based drugs is that they've actually been around for 20 years. And this is the thing, right? So if you've worked within the field, they're not all that new. We understood the biology, but it was the once weekly aspect of it. And the first one was obviously semaglutide or Ozempic. And it happened really right smack bang in the middle of, I forget which lockdown. Was it Delta? Was it whatever? But it happened during that time, where suddenly there was a once weekly. The effect size went into double digits, meaning that the weight loss with these class of drugs had hovered around the 8, 9, maybe 10% mark forever. And then suddenly, bang, this thing, Ozempic, semaglutide, resulted in this 15% weight loss. And maybe coupled with us sat on our backsides at home looking at Twitter. I don't know what it was, but it caught fire. And so now, this is all everyone can talk about, right? And then clearly semaglutide, which is Ozempic and Wegovy, was just the first wave. Yeah. And then now we have tirzepatide, which is Mounjaro and Zepbound, and then a whole host of other things coming behind, now reaching anywhere from 20 to 25% weight loss. And so suddenly we have these tools which are hugely effective, largely safe. And we've never had that before, I have to say, within the field of obesity. And a side effect of this, positive or negative, or however you want to take it, is that suddenly everyone wants to study obesity or develop drugs for obesity. So that, in the past 5 years, has been the biggest change.
Do you think that this wave has taught us anything fundamental about the biology of obesity? The reason I ask is because you say they've been around for 20 years, so we know the mechanism, but now we have millions and millions of people taking them. What have we learned about biology from this massive real-world experience?
That's an interesting question. I have used it. Okay, I think for people within the field, there's no new biology I think we picked up. Genetics has always pointed out that the brain was important for food intake, for regulating food intake, hence obesity, hence body weight. And all these drugs have done is to confirm that, right? Because they all, while they're produced, their gut hormones, they're modified gut hormones, they're produced peripherally, they signal to the brain. What has been interesting is to see people within scientists and actually just the broader public begin to sort of understand it. So two things, that the brain plays a role, and you might think it was obvious, but maybe not to people outside. But crucially, that this has shown us that actually there is a hormonal basis to regulation of food intake. Because whenever I talk about this, I say, look, if you're taking something like semaglutide or Ozempic, you're changing one hormone. If you take tirzepatide, then you're changing two hormones. But ultimately, that's all you're doing. You're changing the concentration in your blood of one or two hormones, and suddenly your feeding behavior changes. And I've been trying to use this as a lesson to try and tell people, so therefore, you can almost consider some obesity, at least, to be some kind of hormonal deficiency rather than a moral deficiency.
Yes.
For that. What has surprised, I think, many within the field has been how effective it's been in terms of the effect size. Okay, some people say they're not surprised. I was certainly surprised.
Yeah.
Anywhere from 15 to 25%. It's ridiculously effective. 5 years ago, would we have predicted that actually we could get close to bariatric surgery levels of weight loss through a drug that was actually broadly safe? No, I don't think we would have, many of us would have sort of put down money on that. Or certainly I wouldn't have.
Are there there's a lot of talk about impacts beyond obesity on addiction, on Parkinson's, that I know a lot of these studies are in progress. Does the basic biology tell us anything about whether these are likely to be successful or not, or have any of the RCTs shown anything meaningful from your perspective?
So from the neurodegeneration, Parkinson's and Alzheimer's in particular, I think the data, looking at the phase two, I think it's in phase 2 or is in phase 3. It's one of the phases anyway, one of the big studies. The data look just speaking to people within the field, the data look good. Now we have to look to see the what exactly is happening, but the data look good. Now the why is interesting. Mechanistically, I don't think any of us know with a capital K. I was going to say M capital K, but I think the current hypothesis is that it is centrally driven and it is some kind of anti-inflammatory effect that these GLP-1s appear to be having. And I think a lot of the stuff that's coming down, for example, the weight-independent benefits for heart disease and kidney disease, there is a weight dependent. Obviously, you lose weight, then you improve your profile for a number of these diseases. But I think the weight independent effects the hypothesis tends to be this anti-inflammatory, which is a kind of amorphous term. But I think that's the current hypothesis that is undergoing. Now, I think it'll be really, really interesting in order to do that. The question about the addiction, however, I don't know. So why does taking these drugs suddenly, if you want to stop smoking or drinking or whatever it is, drugs of abuse, why is it suddenly easier? Right? Yeah, I know. Why is it suddenly easier? Once again, we don't actually know. My guess, and this is just a guess, is that because, look, eating feels nice for most people. This is large way, in part why we do it. And if you take these drugs and they make you feel fuller, I think for some people, they kind of take the edge off how nice it feels. And I just get the feeling, this is entirely opinion, by the way, there's no data to back this up.
Yeah.
But I think because it takes the edge off this niceness, and ultimately everything that's nice ends up in the same part of the brain, different pathways, but ends up in the same part of the brain, that I think it takes the edge off other things as well. Like, for example, smoking or drinking, whatever you used to enjoy, therefore making it easier for you to quit. Let's see. Let's see what happens.
Yeah, I mean, that's a really interesting way to think about it, because there is some like very fundamental psychological principles going on here, right? If you're stressed, people eat, people smoke, people play games, people gamble, people do all sorts of things to
feel nicer, to feel less stressed.
Yeah, that's really interesting. What about long-term effects? What have we learned? A lot of people, there's a lot of data, but there's also a lot of just fear of the unknown because we've never run this kind of experiment at scale before, right? Are we learning anything about, you mentioned that they're largely safe. Are we learning anything about where they're not safe, or is this a free lunch in some respect?
I find it highly unlikely as a free lunch. I think the common side effects are known. Okay,
Yeah.
it tends to affect the gastrointestinal tract because of the biology of the source hormone. The rarer ones, and these include, for example, the anti-inflammatory effects, the effects on addiction. So the side effects can be positive and negative,
Yeah.
but the negative side of the addiction is this rare instances of suicidal ideation. So I just feel that the moment you get millions upon millions upon millions of people taking it, we are going to find
Is it
rare.
going to show up? Yeah.
Exactly, it's going to show up, and then we're going to have to understand it. Right now, I don't. We can't throw the baby out of the bathwater, so to speak. So we do have to understand it, but also accept that it's gonna help so many people. While the drugs have been around for 20 years, as one's weekly injection, one's daily injections broadly, I think so. From first principles, it looks broadly safe. The issue with these once weekly is, and once monthly, if you take the other drugs that are coming in, is clearly the different kinetics, right? And
Mm.
so I do think we will need to keep a watching brief. I do think that these drugs should be prescribed for a long time to come yet, until we understand
Yeah.
what they do.
What do we know about the role that genetics plays in either response or non-response or some of these side effects? I've seen data, for example, PNPLA3, one of the genes involved in MASH liver disease, looks like tirzepatide may not be as effective in certain genetic subtypes. But more broadly, this is probably a very rich area now that we have millions of people. You can combine it with genotype data. What are we learning there?
So oddly, I mean, as a geneticist, you're a geneticist, I'm a geneticist. If I were running some big old trial and putting in billions, which is probably what this cost, I would have got some DNA. Okay,
Yeah.
just saying. Okay, or at least got the ethics. None of the drug companies did. Okay, so they went out, they did the trials, and they found out it worked. And now that it has, because these things have become blockbusters. I think genuinely we can call them blockbusters. They're sort of reverse engineering the process, and so
Yeah.
actually all the major companies are now going back and asking this exact question. And ultimately, it's important for them, right? Because if we can identify the people that don't respond, that's interesting. But the people that hyper respond, and these are likely to end up with side effects and stuff. Or actually, the interesting thing is people that once coming off the drug either don't gain the weight back rapidly or gain the weight back too rapidly. So I think there are a
Yeah.
number of different facets to look at it. And this is going to be really fascinating. I'm not personally involved in some of these studies, but I know that a number of my academic geneticist colleagues who are, clearly, why would these companies reinvent the wheel? I know they have in-house
Yeah.
geneticists, but they're going to engage with people, experts within the field in order to look at it. So it's currently being done, but not
Yeah.
originally. It was not done originally, which was a
Right.
shame in some ways.
My hypothesis on that is that I think the incentive of the industry, probably they want to push for the broadest label possible. But as soon as there's a lot of competition, then that no longer becomes, you want to get there first, so as many people as possible. But now we're in the phase where everybody needs to understand, where does our therapeutic really work and where does it really not? And then how do we position, right, in a much more crowded market than it was 5 years ago?
I think competition is always a good thing. I think actually all of these companies, even though they're in competition with each other, when we're dealing with obesity and all of its attendant comorbidities, it's a pretty big pie to split
Yeah.
up. I think there's gonna be, I think it's it's going to be fine for the drug companies. I'm not feeling sorry for them. I
No.
think they will end up making a lot of money from this, and actually end up hopefully helping a lot of people. What is going to be interesting though, and I realize this is not the genetics element to it, but nonetheless, there's been a lot of talk about it's these drugs. Let me tell you something about these drugs that are interesting. So we live in a country with an NHS, okay, and so clearly the vast majority of us get our drugs from the NHS, whatever drugs we happen to be on. This is the one example that I can think of. Bar aspirin or paracetamol that we go to Tesco's to buy.
Right.
The vast majority of people on these drugs are taking it in this country privately. Now,
Yeah.
that is an interesting phenomenon. I think there are people saying that it's too expensive for the NHS or this or that or what have you. I think this cost is A, I don't think is that expensive. I mean, by drug standards.
Yeah.
B, it's a short-term problem. First of all, because of competition, and that market forces will push down prices.
Yeah.
But crucially, these drugs are going to come off patent. And Ozempic comes off patent in 2032. 7 years, yes, long time, not that long.
Yeah.
The moment it comes off patent, suddenly it's now 150, 200 pounds a month. It's going to drop tenfold. It's going to suddenly be 10 or 20 pounds a month. Then we'll get real, true penetration, I think, of these drugs through the population.
Yeah, that is wild to think about because it is cost prohibitive for most people out of pocket today. But it won't, like you said, we were speaking on this podcast 5 years ago. It's not hard to imagine 5 years in the future this being
exactly right. I mean, this is like exactly the same period of time that we last spoke. Don't say that; it makes me feel old.
Yeah,
but anyway,
I know, me too. Yeah, a lot has happened since then. Well, I'm not going to say last because I always end up with one more question. But I'm curious about other genetic targets, orthogonal genetic targets potentially, because you talked about we've got one or two hormones that you're hitting. Are there other things that big GWAS studies are telling us about other ways that we might be able to tackle obesity, whether it's also in the brain, but through some different mechanism or through some other mechanism? Because there's a few, it's largely driven by behavior and the brain, right? But we can also go after fat storage or other kinds of mechanisms, right? Or what do we know about what else is coming down the pipeline?
So I think there are two. We can almost divide this into two. There are obviously efforts to target the causes of obesity. So this tends to be the food intake stuff, stopping you eating in order to get you. And this semaglutide and tirzepatide would fit in this. And I think there are a couple of things that are not coming online, preclinical stuff that we're beginning to identify. Work that I'm doing, for example, trying to identify new genes or through genetics within the brain. And we have also been the other thing we've been doing is looking and mapping the feeding circuitry of the brains using these spatial transcriptomic approaches and layering on genetics to try and identify new drivers. So that's one aspect to it. The second aspect is the comorbidities, right? Because what that happens is, well, why are there people who are larger, close to obesity? I mean, we can debate whether there is any true healthy obesity, okay, or is it just some temporal phase? But anyway, there are undoubtedly people who are larger but still healthy, and people who are not anywhere close to as large but still ill. And so I think that is the other thing. Can we unpick the lock of that particular area to say, genetically or otherwise, to say, well, why do some people store fat more safely, okay, and others not? And are there any pathways there that we can tweak, not to stop the food intake to begin with, but actually to improve the comorbidities or the likely comorbidities that there are to actually emerge?
Yeah, it's interesting. It's kind of sort of like a metabolic health axis that is different and related, obviously, to intake. But if you've taken that part out of the equation, what remains?
I mean, like some of these new, this is still in some ways incretin-based, but one of the new drugs from Lilly I think it's in phase 3, is something called retatrutide. Now, retatrutide it has the GLP and GIP element to it, so that's fine. But it also targets, it's a triple agonist. It also targets the glucagon receptor.
Right.
Now, the glucagon receptor is everywhere, including in the brain, but actually particularly richly expressed within the liver. And so retatrutide, for example, in addition to the weight loss elements and what we talked about, is particularly good at fatty liver, at actually reducing the amount of fat within the fatty liver. And that is obviously a comorbidity, right? Because if you can prevent that, then you prevent a lot of the other illness independent of your body weight. And so that's a classic example. They've tethered it on to the food intake to sort of maximize the effects. But there are going to be, I think, a host of other targets where you can reduce cholesterol levels or blood pressure or whatever it is, independent of the weight loss.
Yeah, interesting. When we spoke 5 years ago now, almost 6 years ago, I guess, you changed the way that I think about obesity and genetics. In particular, you mentioned this earlier, but viewing it as through the lens of evolution, I guess, is really the way to think about it. I think most people who haven't studied this have a very surface level view of it's got something to do with the way we store fat, the choices that we make. But when we spoke about this, and there's been a lot of work on this in the past few years of really understanding it as we have this evolutionary drive that's been built into us a long time ago, and our world, we're not wired for the world that we live in, where there's an abundance of food. And I think the GLP-1s have proved this in spades, that it's about behavior and it's about the things that our brain causes us to do in this environment that we've really not been evolutionarily trained for. I wonder if you could talk a little bit about that, because I think it's really important for people to understand that the different axes on which this operates and how it's not. The science doesn't point towards it being a choice in many cases. A choice is a funny word in this case, because we have the opportunity to make different decisions every day, and we know that, but our brain is screaming at us to do particular things. And so I wonder if you could just expand on that a little bit for people.
So I think the choice thing is an interesting one. I'll get to that in a second, but evolution first. I think what are the big evolutionary drives? Macro, okay, so you know, from space, looking down. And I think really there are only two true existential traits that living creatures have, okay? And that is feeding and reproduction. You fail at one, you fail at life. Yeah. And so what happens is whatever single cell or multiple cell organisms, okay? And so all of the pressure is always on there to make sure that we feed enough so that we can get into a position to reproduce, so that et cetera, et cetera.
Yeah.
And the vast majority of the pressure that comes from feeding, okay, let's focus on feeding, although it is intimately linked to reproduction, because if you don't feed enough, you won't reproduce. But from a feeding perspective, is that it has stopped us from starving. And that undoubtedly is the big genetic selection pressure that is kept in. And so all of the things that are put in place has meant that if there is spare food, if there is any advantage to one modicum of a percentage of motivation to sort of run towards the antelope or whatever, you know, that would have been selected for. This is not to say there hasn't been any selection pressure on being too large, okay? Just fewer. Clearly, if you are too large, you'll get eaten by the saber-toothed tiger, okay?
Right.
And I think a classic example of being too large is the peacock, okay? How large can the feathers get before they get eaten versus I need a mate? And the peacocks I would say peacocks are probably
pushing the limits.
Exactly, pushing the limits. But so I think we have to consider everything that we and how we respond through that prism, right? Yeah. And for the vast majority of time, we had not enough food, even through agriculture, even 50 years ago, all right? Where this is why people say, well, we were skinny 50 years ago, which is probably broadly true. That we have brains, put simply, that are designed to face a feast-famine scenario, which is how we were. Sometimes we had the antelope, sometimes we didn't. This concept of a feast-to-feast environment, which is what we're living in, is a contemporary one. And so hence we have a brain that is maladjusted for a feast-feast environment. And then throw onto that the genes, right? Because you say, well, but not everyone is fat. And that's true. Obviously, that's true. So I think what has happened is our genes haven't changed, clearly. But in one environment where we have not had this constant food push from the environment, we're fine. But suddenly we have Uber Eats and suddenly I don't even have to go to the shop. I can, from my couch, I can call food, right? Someone can walk the food to me. Then this availability has suddenly exposed people with susceptible genes that are more likely to the one example I would give is this, actually. When I first moved to the UK, this would have been the early 90s. Shops did not open on Sunday. Yeah. So if on a Sunday in 1990, whatever, okay, I wanted a bowl of cereal, but I didn't have cereal or milk, tough. Right. I had to wait till Monday, right? Because what am I going to do, right? So I could not deliver. Even if I wanted it, I couldn't deliver it. Today, we can. And I think that's the difference, where suddenly my desire for cereal can be instantly, you know what I mean? You know, almost saying, yeah, you can now get it. So I think that from an evolutionary perspective, we need to understand, which is why fixing the environment is, from a policy perspective, is crucial, together with drugs and education and other things, is crucial to actually trying to fix the obesity epidemic to begin with.
I think you nailed it. I mean, my reflection on that is really similar to yours. I'm from the US, as people can probably tell by the way I talk, but I live in the UK. I spent 3 months in the US last summer, and even the difference actually between Cambridge, if I drive from one side of Cambridge to the other, I don't pass a single fast food restaurant. I can make it home without Bojangles and McDonald's getting in my vision. In suburban North Carolina, a 15-minute drive, you will pass 15 different fast food options. And it's even interesting to just think about the difference between those two environments that I was in. And the UK has its challenges, but that opened my eyes a little bit to how different actually to quote unquote Western environments with Western diets still can be. And even in the US, you go to plenty of places where you don't have that phenomenon, but just driving by them every day, the option is always there to pull into the drive-through and grab a chicken biscuit. Yeah.
I remember this. I remember what you're talking about, the choice option. So
Yeah.
the choice option is this, right? Where let's go to your example, where you're driving past, you see the chicken shop, you see a McDonald's, and you may choose to turn into the drive-in or whatever you're doing. But my take on this has always been this. It's a nuanced take, okay? That we do not gain or lose weight overnight. Now, any given meal, no matter how large, is not going to change our weight, right?
Yeah.
Our weight is the function of thousands of feeding events that have happened over the past few years. But I think where genes come in, your genetic hand of cards, is that it makes some people a little bit less likely to say no. So 5% is probably the kind of range we're looking at. Now, when you take 5% over thousands of feeding events, that's hundreds of thousands of calories. So while each individual meal is a choice and you can say no, in casino terms, the house will always win. Over a lifetime, if you say 1 out of 20 times, you say, I will turn into to the shop, that's hundreds of thousands of calories different over a few years.
Yeah, I think that's a great way to put it. It dovetails to one of the questions I had for you, which is around you and your colleagues, people you worked with, have studied lots of rare and ultra rare syndromic forms of obesity, often affect children. People may be familiar with these kinds of cases. That's one end of a barbell. On the other end, we have common complex obesity we've been spending most of the time talking about. I'm curious whether this is a continuum and what those rare and ultra rare and the genes we've identified there have to teach us about common obesity and vice versa.
I think you're right. When we first started, we sort of siloed them into monogenic rare causes of obesity with large effects, but true for actually any disease, versus polymorphisms with each having a small effect. Undoubtedly, we now know it's a spectrum, okay, where there is a whole flow, because for two different reasons. First, and this is stuff we knew a little while ago, that when you look at the GWAS genes that actually appear, or at least the genes closest to the loci that actually appear, a lot of them, when you look, also appear as rare monogenic loss of function mutations. So we understand that actually, if you have severe mutations, deletions, and things in some genes, you end up with severe phenotypes, whereas subtle polymorphisms result in subtle phenotypes. So clearly, similar pathways are involved, just to differing degrees of perturbation. Now, what has recently happened, and because is XWAS, this so-called exome-wide association studies, where you take whole exome sequencing at scale, and instead of using each individual SNP as the statistical moiety which you're testing, you sort of do a gene burden test. You take all of the changes, rare or common, collapse it into the gene. So this XWAS studies. And what has happened there is actually the moment you do whole exome sequencing and you can find all the unknowns, shall we say, of these genes and rare causes, suddenly you're seeing far more nuance in terms of what do we mean by population level changes, because we have done XWAS studies and identified genes, rare. In UK Biobank, we found in this one gene called bassoon, okay, so we published last year, where we found 65 individuals in UK Biobank, with protein truncating variation, 65 out of 500,000. But the effect size was 4 BMI points. It was at the population level, the largest we have seen, but rare. And so that is what is beginning to inform us, these new technologies, that actually these silos make no sense. And I think we have to sort of study it. We can use both. In fact, what we do now, actually, because of our access to these whole exome, whole genome sequencing, is we will start with the rare loss of function mutations, bypassing the problems with GWAS in the past, where we didn't know what gene was evolved and it was too small. Whereas you self-identify the gene because of their loss of function, but then go back to using GWAS and SNPs to ask, are there subtle variations that then give you a more subtle phenotype? And so it's definitely a spectrum now. And this whole concept of XWAS and whole exome sequencing at scale, that has really, really changed the pace at which we're identifying new genes for all traits. I'm interested in body weight, but for body weight in particular.
What is whenever we're doing science, there's like where the light is shining and where we can look, and then there's all the darkness. What's the next frontier that you think about? We don't have the tools today, or we're just getting the tools, and so we don't actually know the answer to these key questions, but they're on the horizon. Or if it's interesting, ones that are not on the horizon, just because we have no way to answer the question or no way to understand. Where's the darkness in our knowledge right now?
Well, let's start at the hinterland of the darkness first. Okay, just
Yeah.
as we push ourselves outwards. The new genetic technologies at our beck and call mean that we can now do reverse genetics using pardon me, forward genetics, not reverse genetics, forward genetics using humans. In the past, you had to go and you had to knock out a gene in a mouse, right? And then find out what the phenotype was, and then sort of back and ask, well, is there a mutation in a human? But
Right.
now, because we are in a situation where genetics has been so democratized, where actually the vast majority of the large super cohorts that are around, UK Biobank, et cetera, they're all open access now. And so you can now go and any given trait, any given gene, and says, well, is there a loss of function in gene X? What may a phenotype look like? And then work out the biology from there. And so hence, I think that gives a lot of room to identify new, as long as it's been measured in the cohort. Okay. So this is why there's the shadow. In terms of the unknowns, I mean, I think there is still, and people always ask me this question about epigenetics. Okay. Because obviously is that I think there is a lot to look at there. The main issue with epigenetics, as everyone knows, is it's a little bit volatile. It's not that volatile, but it's volatile, but it's also organ specific. And so for us who study feeding behavior, and that's brain, there's an accessibility to the source organ. But nonetheless, I think it's still going to be important for us to understand in obesity or whatever, what changes have occurred. Beyond that, I don't know. I think the genetics.
Hard to know.
I know. I think we're still chipping away at these large cohorts. So I think better phenotyping is always going to be key, because I think we've got the genetics. The price is only going to go down.
Yeah.
We already have whole genome sequencing. We're only going to get better, I think, at doing this cheaper and quicker. The phenotyping of a patient group at scale will always be the step that is absolutely crucial to make the genetic links, but also it's very difficult to scale effectively, at least yet. So I think that is probably the other side of the thing that we have to really push the technologies to phenotype large groups of patients at scale better.
You published a second book in 2021, I think it was. That
it's a COVID book, yeah.
It allows you to probably focus on things that you didn't have time for before. It was it's a controversial topic, not in the kind of Hollywood sense, but the premise of the book is that calorie counting in weight loss is actually really a red herring, right? And the physicists who look at energy expenditure and those sort of things will probably say, like, what does he mean? Calories in, calories out. But there's a lot of nuanced arguments in there about it's not just about the quantity and an energy density sense, but it's about what is that coming from. I wonder if you could talk a little bit about that. How did you get interested in writing it, and what's the core science and premise behind it?
Okay, so I got interested in it because I'm interested in food intake, and I think it was around a time where there was a lot of discussion about calorie labeling, and now actually we have calorie labeling in a lot of places, and so it just got me thinking about the concept of the calorie, and actually how do we improve our diets? This is why I was interested in it.
Yeah.
Now the issue with whenever I talk about this, you're right. The physicists understand what I'm talking about. The gym bros, the guys who wear tight t-shirts with big buff, they really don't like me saying this. Look, I'm not anti-physics, and I understand that 200 calories of chips is twice the amount of chips as 100 calories of chips. But I guess so is 200 grams of carrots, twice the portion of 100 grams of carrots, and no one is trying to compare 200 grams of carrots to 200 grams of chips. Ultimately, that's it, right? Where calorie is a one-dimensional measure. It measures how much food is actually there, but it doesn't actually tell us anything about the nutritional content of the food. That is why I think they don't count. That's the first thing. The second thing is we eat food and we don't eat calories. Yeah. So what happens is we eat, yes, a calorie is a calorie. Once it's in you, within your mitochondria as a little poof of energy. Okay, yes, you're absolutely right. That a calorie is a calorie. But the process of getting a piece of food, be it a celery, sugar, or a piece of steak, into you and into that poof of energy requires, depending on what you're eating, differing amounts of energy. And
Right.
so you have to put in energy to get energy. And the more complex or the more fibrous or the more thingy, the more energy it uses. So the food matters,
Yeah.
even when once the proof of energy in you, that is equal. You're
Right.
absolutely right. But we eat food, we don't eat calories. So
And
broadly
that's why
speaking, that's what the book is.
and that's probably why the physicists like you, because you've made it clear that like we're talking about the whole energy in the system, right? Not just because you're exactly right. A carrot takes, I mean, we've all heard these stats before, but I think a carrot may take more calories to digest than it actually contains, right? So it's like one of these nearly neutral kinds of foods, whereas a chip will just fly through you, right?
Exactly.
Yeah.
Exactly. Yeah.
Yeah, that's interesting. I'm wondering about the implications on how we as a society design policy and food systems. What's like, if you follow that premise, what does that lead us towards?
Okay. So I think if we follow that premise, what is the aim? The aim ultimately is to get the population healthier. Okay. So to that, I hope that's everyone's aim. So let's assume that's the aim. And if we make a second assumption that part of that aim has to be to improve the diets of the population. How do we do that? And I would argue just by putting calorie counts on a menu is not going to do that. So I think what we've got to do is to try and sort of label, because I don't think it's an information issue. Because if you go into a supermarket and pick out a prepackaged food and look at the back, all the information is there.
Yeah.
I mean, in very small font and in low lighting, it's just terrible to read, but all the information is there. It's not like there is lack of information. So putting more information on there, I don't think is helpful. I think what we need to do is highlight the right bits of it or the more appropriate bits of information. To my view, and this is just my view, I think what we have to do is to highlight nutritional density. So for example, how much protein is there in this particular food? How much fiber is there in this in particular food? And you're right, don't do it in grams because I don't know what 20 grams of protein looks like, right? Whereas I think there should be the traffic lights that exist at the moment, traffic light signals, rather than doing it for calories, rather than doing it for some other things, do it for the amount of protein in the food, the amount of fiber in the food, the amount of maybe saturated fats in the food or whatever, or free sugars. I think that there are items that we can pick out that will improve the quality of the food that we're actually eating. And I think that is what should be highlighted rather than calories, which is easier. I appreciate that because it's a number, it's a single thing.
Yeah.
But that doesn't mean that it is the best way to approach this.
What about the great debate going on about processing, ultra processing? What are your thoughts on that? Because this is a lightning rod at the moment.
It is a lightning rod. So just briefly, ultra processing is most foods we eat, aside from if I walked out into the garden and plucked an apple off a tree, has been processed. Okay, cooking, fermentation, these are all processes. Ultra processing, quote unquote, are industrial processes that we can't replicate in a domestic situation in our kitchen, for example. And once again, I'm a nuanced person. For my sins. And there is a nuanced argument to it. And whenever I say this, people think I am funded by big food. I am not. Okay. But it's interesting. My issue with the concept of ultra processing is it's too blunt a tool. It's too broad a church. It includes too many things. Now, there are certainly foods, and we talked about some of them that you may be driving past in North Carolina or somewhere, where I think it's uncontroversial that actually we probably shouldn't be eating as much of. Okay. And I think most of us can agree on a key group of foods. There are, however, a huge number of foods that lie in a gray area. Let's take one example, yogurt. Okay. Now, if you have yogurt, just a white natural yogurt, that is processed, it's been fermented or what have you, it's not ultra processed. But if you buy certain brands of yogurt, where they have, for example, put crunchy stuff in or maybe a little bit of jam in or something in order to flavor it, suddenly the whole yogurt becomes ultra processed. Now, a teaspoon of jam into it suddenly makes the whole thing ultra processed, and this is now suddenly evil for you. I don't know, right? It's very, very different from something which has been constructed from molecular basis from recovered something meat product, right? And so, and the other thing is probably supermarket bread. Now, taste aside, I realize the bougie bread down at your bread, sourdough bread is delicious. Okay. But it also costs £5 a loaf. right? Now, if you go to the supermarket and buy your 30 pence, pack of white loaf, yes, it tastes not as good. Okay, fair. But is it bad for you in a sense where suddenly it's going to kill you? And bread is flour, yeast, a bit of salt, some water, maybe a little bit of sugar, maybe. And in a supermarket loaf, maybe some emulsifiers and things. And so that's my problem with ultra processed. I think it's such a broad church. Yes, when you do the
Yeah.
association studies, sure, if I have too many chicken nuggets, it's not going to be great for me. I don't think that's
Right.
a big surprise. But I think we need to be more precise. And I
Yeah.
think ultra processed is too broad a church. That's my opinion.
Yeah. Yeah. I think it's fair. And it strikes me as a challenging question to answer because for two reasons. One, running RCTs, randomized control trials, in something like this
It's
is
nearly impossible.
crazy hard. But I think there's a second factor, which is because we run crazy hard RCTs in gene therapies and things like that, but there's a clear financial motivator. Someone is developing a new therapeutic, and I'm not sure who benefits from settling this question of labeling the different degrees of ultra processed. I don't know if there's even any entity that has the financial incentive to run the big RCT to answer that question, right? I'm not sure who, and it's not, and this is not a conspiracy. It's just that it's such a broad question. It's so important to our health to answer, but it's it's an academic level question that then becomes insanely expensive, right? How do you measure what somebody eats constantly all day for tens of thousands of people? It's just hard.
But I do think it is important, at least for us to think about what about nutritional quality, and ultimately
Yeah.
this is what we're talking about, right? They're going to be many ultra processed foods are going to be awful for you, or you shouldn't eat a load of. Whereas there are actually some which are just caught in because of some
Yeah.
technicality that they're caught in, and in a time where there is a lot of inequity and food insecurity that matters. Okay, it may not matter to you or to you or me because we have choices and we can do what we want and we probably need to think a little bit harder somewhere else.
Yeah.
But if you are within the bottom quintile of socioeconomically and you have very few choices because of a lack of cash, a lack of time, then we shouldn't be food shaming people. What we should be doing is trying to equitably improve the food environment rather than taking food that actually is fine for you and sort of wrapping it up in some judgmental statement. So I agree that it is very expensive to answer in certain aspects, but I think in other aspects, I think we do need to really think harder, and it's
Hmm.
crucial about equitable nutrition, for across all of society.
Yeah. Do you have any ideas on how we might be able to settle that question scientifically?
I think all the information is there because when you manufacture a food, this is obviously not going to the butcher and buying a piece of steak. It's a very different thing, right?
Yeah.
We're talking at the moment, on average, 50% of our calories come from prepackaged food, and that's fine. I'm not look. It may be some people might think that's an awful thing. Maybe we need to improve it, but ultimately, it's kept all of us alive. So I think the information is there, and I think what we need to do is how do we leverage this information to make sure that we incentivize companies to make the healthier options. Now, I'm not talking about converting a frozen lasagna into a lentil. I mean, because that's not what I'm talking about, right? But I'm talking about sometimes in life, yeah, I'll eat an apple or a banana and I'm very happy with that. But sometimes in life, you need a chocolate bar. Okay,
Yeah.
sometimes life demands a chocolate bar. The question is, can we take a chocolate bar, because sometimes there are chocolate bar moments, and make a healthier chocolate bar? Can
Yeah.
I put more fiber in it? Can I put more protein in it? Can I, sort of Trojan horse in some more minerals, so that when you are in the chocolate bar mode, you can have a healthier chocolate bar. It's
Yeah.
never going to be healthy, quote unquote. But I think that is the kind of thing that we need to be thinking about.
Yeah. Yeah. I think it's a very, there's some common sense principles to this as well, right? Like I think about the foods that if you left them outside, how long would it take them to mold, right?
Yes,
There's some of these things we could just say, okay, we've all seen the McDonald's cheeseburger that takes 3 months to mold. So
yeah,
we could probably agree that
correct.
there's something going on there, but everything else is more nuanced.
Yeah, I think
Yeah.
so. And I think we need to discuss this in a non-hysterical fashion. And we need to discuss this where, and I have this discussion, say, with you, okay, that no one is saying I'm funded by big food because I'm trying
Yeah.
to defend some elements of it. You know what I mean? It's just, it's so non-hysterical discussion is how we're going to get into to at least fix some of these more knotty problems.
Yeah, totally. As we wrap up here, I'm curious about what's next for you. You've had a prolific last 5 years, multiple books, tons of great research work. What are you most excited about now? Personal, professional?
I think I look, I think it's two things. I mean, first, from the genetics perspective, actually, with the prospect of all this whole exome sequencing data, we are actually identifying new genes. Rare, yes. So I'm not talking common causes of obesity, but actually it's beginning to sort of open up the map a bit more. And I'm really, really interested in doing that. So that's the first thing. The second thing is we are now getting, with the technologies available, we're now having the ability to, all of the feeding circuits prior to the past few years have all come from mouse studies for obvious reasons, okay? But now, because of technologies being available and the availability of the right donor samples, we're able to do now this mapping within the human context. And so I'm mapping various parts of the brain that are crucial for sensing peripheral hormones, for example.
Yep.
And I think that is probably, in the next few years, those are the two things I want to do. Try and find more genes to fit into the jigsaw, and also then map these onto the human context.
Yeah. One more bonus question. How do you find writing books? Is it easy? Is it hard? Do you get the writer's block? I'm just curious because some people write one and they say never do it again, but you've written two, so I'm wondering if there's a third in the works, and also more generally, like, how do you do it? What's your process?
So there is a third rattling around my brain. I did a couple of years, well, when I turned 50, in lieu of buying a Porsche, I cycled from Land's End to John O'Groats, so it's about 1,000 miles. And I did it with a couple of glucose monitors on me, okay?
Yeah.
And so there's a story to be told there. I just don't have the time at the moment. And because the first two books I wrote, okay, one of them was COVID, so you can't count that. The first book I wrote in the evenings or on weekends, and I was just a slightly less busy person now. So the idea is there, but I want to do it. Now, how do I find the process? It's very, very different from writing a paper. And you might think, but it's still writing. The thing about writing a paper or writing an academic writing, it is very, very heavily referenced and very dense. So each paragraph will contain multiple facts or points or data points. Yeah. It's very different from writing a popular science book, because I had to learn how to what did my publisher tell me? Let the text breathe. I said, what the hell does that mean? And so in other words, you have to find a facts per 1,000 words. Okay?
Right.
How many facts should you be delivering per 1,000 words? And actually, then it becomes easier to write, because then the way that I did it was, okay, I have this fact. Okay, the brain is important for this or something like that. And I have a flight to Edinburgh, and I know it's going to take me 55 minutes. Can I produce 500 words on a flight to Edinburgh, which incorporates this fact in a sort of interesting fashion? And so I'm only thinking one fact, and I've got to tell a story, wrap up the fact, and deliver the fact. That's a very different writing challenge to saying 500 words is an abstract, okay, and delivering a whole paper in that thing. That takes far longer to write, and it's a different process. It's a different process.
Yeah, that's super interesting, though. I love that way of thinking about it, and it's probably simultaneously challenging if you're in that academic paper mode, but it's also kind of freeing. It's just, you can write and write and write for 500 words, 1,000 words, and then you edit and pare back and make it a little better. But there's no pressure to nail every sentence, right? The first time. Yeah,
exactly. So it's just a different type of. I'm not belittling. Okay, it's facts.
Yeah,
but it's just who is reading it, who's reading the piece. When I'm reading a paper, I want facts.
Yeah,
give me the data. When I'm reading a book, I'm not looking for. I'm looking for something different.
No, and I remember when I first started my PhD, the way that I learned is I would pick up a paper, I would read the first sentence and go, I don't know that word. Google it, figure it out. Once you understand the sentence, go on to number 2.
Go to the next one.
You don't want that in a book, though.
No.
You don't want that in a book. Yeah. Well, Giles, thank you. This was really fun. You're an incredible guest to have. So thank you so much for coming back for the second time. I'm sure we'll make it a third, but hopefully a little sooner than 5 years. Things are moving very fast.
Thank you for having me on.
Thanks, everybody, as always, for listening, and we will see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah who produced the show, and James Pierce from Selected Frequencies who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.